Hello everyone, welcome to my channel. In today's video, we will discuss an important topic in microbiology and antimicrobial resistance, inducible clindamycin resistance, also known as the D-test. This concept is especially important when dealing with infections caused by Staphylococcus disease. Let's begin with the basics. Inducible clindamycin resistance is related to a group of antibiotics known as MLSB, which stands for macrolides, leukocytes, streptogrammin B. These antibiotics work by inhibiting protein synthesis in bacteria. They bind to the 50S ribosomal subunit, preventing bacterial growth. However, in gram-positive bacteria, especially Staphylococci, resistance can develop. This resistance is mainly due to methylation of the ribosomal target site, which is controlled by genes. If we further dive deeper, there are three main mechanisms. First one is target site modification, that is, ribosomal RNA methylation. Genes encodes methylase enzyme, which attaches one or two methyl groups to adenine residues in 23S ribosomal RNA, lowering affinity but ribosomal subunits to MLSB antibiotics. Second is antibiotic efflux. This is mediated by MSR genes. Last one is enzymatic inactivation. Genes like ERE and MPHC encodes for enzymes like esterases and phosphotransferases, causing resistance to macrolides. Similarly, genes mediate resistance by producing nucleotidyl transferases. There are two main types of MLSB resistance. First one is constitutive MLSB resistance. In this type, resistance is always present regardless of any inducing agent. During lab testing, bacteria show resistance to both erythromycin and clindamycin, so these antibiotics are ineffective in treatment. The second is inducible MLSB resistance. This is more tricky and clinically important. Resistance appears only in the presence of an inducing agent. Erythromycin acts as a strong inducer of the gene. In laboratory testing, the bacteria may appear resistant to erythromycin, but susceptible to clindamycin. However, during actual treatment, clindamycin resistance can be induced, leading to treatment failure. So why do we perform the D-test? Because standard susceptibility tests may give false susceptibility to clindamycin. The D-test helps us detect hidden inducible resistance, ensuring proper antibiotic therapy. Now let's look at the procedure. First, isolate gram-positive bacteria from clinical samples. Perform a lawn culture on Mueller-Hinton agar. Place antibiotic discs according to CLSI guidelines. One erythromycin disc 15 micrograms, one clindamycin disc 2 micrograms. The discs should be placed 15 to 25 millimeters apart. Incubate the plate and observe the results. Now, how do we interpret the results? If a D-shaped zone of inhibition appears around the clindamycin disc near erythromycin, this is called a positive D-test. This indicates inducible resistance. In this case, even if clindamycin appears sensitive, it should be reported as resistant along with erythromycin. To summarize, inducible clindamycin resistance is an important mechanism in bacteria. It can lead to treatment failure if not detected. The D-test is a simple and effective method to identify this resistance. Thank you for watching this video. If you found it helpful, don't forget to like, share, and subscribe for more educational content. See you in the next video.